This presentation I will be using the smallest pen size possible. Be sure to watch in 1080p or full screen if you would like to see better, and if it sucks, I'm sorry. But for the main theme of regulating glycolysis is all about the sites where we have an irreversible reaction taking place, because if you have a combination of reactions and some of those are reacting in equilibrium, well, it's the ones that are irreversible that really determine the directionality of your reaction and the rate, right? Because we've already gone over like rate limiting steps. And for the skeletal muscle, this is a concept that you're probably already familiarized with to some extent, is the concept of feedback inhibition, which is where the end product of our reaction actually inhibits its own production. And something that we've already really talked about, but it really plays a role, especially in metabolic regulation and specifically glycolysis, is the energy charge. And I've already made a video kind of explaining this in detail, but for the purposes of this, let's just looking at this from 30,000 feet, from seeing the bigger picture here. This is based on the ratio of ATP to AMP. So as we're about to see, one of the main things that we produce in glycolysis is ATP. So ATP is obviously going to act through negative feedback mechanisms to allosterically, so we're thinking quaternary level enzyme structures, inhibit the reactions that they're taking place. And AMP isn't really so much an activator as it is the fact that AMP will block ATP from getting to his allosteric site. A good question that people tend to ask is why we don't use ADP, and because of reasons that I'm about to explain, the ADP can be used by an enzyme named as adenylate kinase to generate ATP, and this is used very, very rapidly. So ultimately, ADP doesn't really exist. As once we start to reach equilibrium, we'll have, very, very high concentrations of AMP and very low concentrations of ATP once we've used up a lot of our energy. So because of the use of adenylate kinase, ATP and AMP are the initial products that we get from ADP. Ultimately, ADP, if we're in an extreme situation, will not exist, which ATP and AMP is our energy charge. So we don't really worry about using ADP as a metabolic regulator. Okay, so for the enzymes that we're ultimately going to be talking about are the ones that are irreversible in glycolysis, and you should probably know what that is already. I'm going to list 3 of them, and hopefully you can guess what they are. So hexokinase, phosphofructokinase, and then pyruvate kinase. Something we're about to find out later is that with hexokinase, it must stay in pretty close connection with phosphofructokinase, and this is because of the fact that it is the first step in both glycolysis and ultimately the production of glycogen. So glucose 6-phosphate acts as the allosteric inhibitor, and because they are connected not just in space but in terms of like sequences of chemical reactions, hexokinase and phosphofructokinase tend to kind of stay in close proximity and communicate with each other. So 3 things I want to say about phosphofructokinase is that it is the primary pacemaker of glycolysis. So why is phosphofructokinase the primary pacemaker and not hexokinase? Well, that's because phosphofructokinase has a specific step, and this is the only job that phosphofructokinase has is in glycolysis. But hexokinase plays roles not only in glycolysis but in the production of glycogen and other things as well, as we've already seen with. And for phosphofructokinase, ATP is its actual inhibitor. And lastly, if you have relatively low pHs, meaning that we're doing a lot of fermentation going on, that also will inactivate phosphofructokinase to slow down the production of acid as well with a negative feedback loop. For pyruvate kinase, it actually has 2 different types of allosteric inhibitors, one of them being ATP and then the other one being alanine, and I'll explain why for that in a second. So the reason that alanine is used as an allosteric inhibitor is because pyruvate is actually an intermediate in alanine synthesis. So pyruvate kinase doesn't just play a role in glycolysis, and this is why phosphofructokinase is the main regulator of glycolysis, because hexokinase plays roles in other metabolic pathways, as does pyruvate kinase, but phosphofructokinase is unique to glycolysis, so obviously that's the main point we want to have in regulating just glycolysis. Okay, so that's it for the skeletal muscle. Now I'm just going to talk about the liver, and the liver actually has, it does to some extent also play a role with feedback inhibition. I'm not going to say that that's something strictly related to skeletal muscle, but the thing is with the liver is it has such a constant supply of blood and things moving in and out that I almost want to think of the liver, chemically speaking, as like an open system, whereas skeletal muscle is kind of like a closed system. So the enzyme involved, basically the hexokinase of the liver, if you could think about it, is called glucokinase, and what a glucokinase is, it's something known as an isozyme. Now, an isozyme is not a isotope or isomer version of an enzyme. It's a enzyme that has a completely different amino acid sequence, but it does the same job. There's more than one way to skin a cat. There's more than one way to make a kinase enzyme that's going to act on a 6-carbon sugar. So glucokinase has an incredibly high Km, which means that its affinity for its substrate is actually a lot lower than that of hexokinase. And the reason why, like I said, is that the liver is an open system, and the liver has blood moving through it. Everything that you eat must go through your liver first before it enters your bloodstream. And so if your liver had an extremely high affinity for this stuff, it would be making all this stuff and hogging it all for itself, whereas your brain, your heart, your lungs, all your other organs, they need to have glucose as well. So this high Km makes sure that only whenever we have a lot of glucose available is it safe to do this. And this is one of the mechanisms we have of just conserving it so that other organisms can have, or other organs can obtain the sugar that they need. And like I had mentioned previously, this is also plays roles in not only fatty acid synthesis, but the production of glycogen, which your liver is really good at. So for the phosphofructokinase, I'm just going to branch this out into the activators and the inhibitors. For inhibitors, we have ATP and citrate. Depending on how much general biology you've taken, citrate is just the deprotonated form of citric acid, which is why we call it the citric acid cycle. That's where that comes from. For an activator, what it actually has is something known as fructose 2,6-bisphosphate, and we'll talk about that in a second. I don't know why I always capitalize the bis. The bis should not be capitalized. So how does fructose 2,6-bisphosphate work? Well, one of the things that it does is it is going to totally block ATP's binding site. And obviously, if you're blocking ATP's binding site, you're going to actually increase phosphofructokinase's affinity for fructose 6-phosphate. For some reason, I thought the word substrate would be shorter, but it's not much shorter. The last enzyme we have to talk about is known as L-pyruvate kinase, the liver pyruvate kinase. So both the L and then the M for muscle, those are isozymes. Okay, so the way that we control this is by phosphorylation. And so let's just say, hypothetically speaking, that you have low blood sugar, you want to conserve as much as you can for the brain, and you have that whole glycogen cyclic AMP cascade happening. Well, that's going to result in the phosphorylation. And in this context, phosphorylation is actually going to be inhibiting L-pyruvate kinase. Phosphorylate L-pyruvate kinase, we're inhibiting it, and if we dephosphorylate it, we're going to be activating it, is what an isozyme is, because I just feel like that's something I should probably write down because it's important. So we have different amino acid sequence, but it does the same reaction.